Next up, I'd like to introduce Bani and Jolie from the CSIR-IGIB Institute in New Delhi. And you should be able to share your screen now.
Am I audible now?
Yes.
Yeah, I'm so sorry about that. So, hi everyone, good morning, good afternoon, and good evening. So, I am Bani. I am a researcher here at CSIR-IGIB in India. And I'm a computer science engineer by profession and currently pursuing my PhD in the field of computational genomics. And for the past 2 years or so, my lab has been working on sequencing and analysis of SARS-CoV-2 genome isolates from India, as well as the analysis of publicly available SARS-CoV-2 genomic data. And now we are moving on to other infectious diseases such as monkeypox. So today I will be talking about what we know about the genetic epidemiology of monkeypox from the genomic data that was available in the public domain, and the insights from the data that we got from analyzing the data and the surprising results that we observed. So the way the world has been dealing with infectious diseases recently has how the world has been dealing with infectious diseases lately is now been changing rapidly. And this is mainly because of two things that I'll be talking about in my next two slides here. So compared to the traditional ways of doing microbiology, the past decade has also seen genomics making its way into the field thanks to the advancements in next generation sequencing technologies. And it is today possible to collect samples, process them, sequence them, and analyze the genome of the pathogen that causes the disease, and also to characterize the pathogen genomically. And the second thing is that viruses, like all organisms, they evolve by accumulating mutations in its genome. This rate of mutation, however, differs between different types of viruses, and multiple studies have now shown that depending on the genetic makeup of a particular virus, this rate differs such that, for example, single-stranded RNA viruses have a higher rate of mutation as compared to double-stranded DNA viruses. By looking at these mutations in a particular pathogen or a virus, we can trace the spread of the disease that it causes and also check the evolution of the virus as the virus continues to spread among hosts. So coming back to the agenda of today's symposium, here is a brief timeline of monkeypox disease outbreak that occurred in 2022. So the outbreak was observed in May 2022 when cases from outside of Africa, where the disease has been endemic for many years, was observed in people that had no travel links to any African region. The epicenter of this outbreak was found to be Europe, and by June 2022, like within a month, more than 1,000 confirmed cases of monkeypox infections were reported to the WHO. So that by as on August 3rd, 2022, WHO declared the 2022 outbreak of monkeypox a public health emergency of international concern. And as of today, that is in September 2022, there are approximately 60,000 reported cases of the disease from over 100 countries and even 19 reports of deaths in patients. So the monkeypox virus is actually a double-stranded DNA virus having a genome of approximately 200,000 bases in size and having approximately 190. And recently, the monkey isolates from across the world have now been characterized as the grade one that is referred to as the and clade 2, which is two sub clade 2A and clade 2B, something that was previously referred as clade. The clade 2 monkeypox genome the human transmission of virus, which is why this clade, the HMPXV1 clade, it further contains different lineages that have been starting to get assigned to the scales of the outbreak. This lineage of this entire cluster is really contains sequences from the 2017 to 2019 outbreak of monkeypox as well as the 2020 outbreak of the disease. So, if we talk about how genomics has been accelerating the how we have been looking at outbreaks of disease and pandemics such as the COVID-19, this rapid sharing of large number of viral genomes in the public domain has enabled governments and institutions to make public health decisions that have been driven by genomics. And as of today, there are over 1,600 complete genome sequences of the monkeypox virus that are available publicly on GISAID. And the number of genomes that are being deposited to GISAID and the number of countries depositing viral isolates for the monkeypox virus are only increasing with time, which enables us to understand the epidemiology of the disease by looking at the mutations that are occurring in the virus, by tracking the lineages that are prevalent across different regions, and creating phylogenetic builds, and then ultimately sharing all of this data publicly to enable better decisions that are driven by genomics. So going by this, when we analyze publicly available data for the HMPXV1 genomes from GISAID, we found that genomes for the monkeypox virus mostly clustered into two major lineages. The first one being the B.1 lineage, which is a which is a descendant of the A.1.1 lineage. This cluster comprised 547 genomes that were available on GISAID at that particular time, and mainly comprised of genomes from European and North American outbreaks. So the 2022 outbreak, if we talk about in lineages, is mainly characterized by the B.1 lineage. However, we also found another cluster of genomes, which is shown here in red, which was the lineage A.2, which at that particular time contains 6 genome sequences from GISAID and is a descendant of the lineage A. So, what is this A.2 lineage? So, this is the phylogenetic tree of the A.2 lineage genome. Genomes in this cluster, so in this cluster, there were 9 genomes from 6 unique clinical isolates that were present on GISAID and were collected or isolated from 3 separate countries. And the earliest genome collected in this cluster actually belongs to Texas, which was isolated in the month of July in 2021. All Indian isolates that were present in GISAID at that particular time also were found in the A.2 lineage, and they were closely clustered with this genome here that was sequenced from Florida. And the samples from Texas, Virginia, and Thailand actually form separate subclusters on the tree of the A.2 lineage. Coming to the origin of the cluster, the TMRCA, or the time to most recent common ancestor, was actually found to be 25th June 2021. And when we talk about what was the travel history of the patients, so most of them had a history of travel to either the Middle East, including patients that belong to India, and in USA and in Texas, and talking about the Texas and Thailand genomes, they were actually isolated from patients who were travelers from Nigeria. So when we looked at the different mutations that were present in A.2 cluster and compared them with the mutations that were present in the B.1 cluster, we found that 16 genetic variations are there in the genomes of A.2 cluster, which are not found in any other lineages. 9 of them were non-synonymous mutations, such that they introduce a change in the amino acid sequence of the genome. 3 were synonymous and 1 was a stop gain variation. And we also found a 3 amino acid deletion in the gene OPG174. So we then proceeded to calculate the nucleotide substitution rate for the A.2 and the B.1 lineage, and we found that the A.2 lineage actually had a lower rate of substitution per site per year, which was approximately 10, which is much lower compared to what we found for the B.1 lineage, which had a substitution rate of 22 substitutions per site per year. Although we do realize that this calculation was actually full of limitations, considering that only a small number of A.2 genomes are actually available currently, so that these estimations may not be very accurate. And this can also be caused by the rapid spread and parallel evolution of B.1 genome, which can lead to some inaccurate estimations of the mean nucleotide substitution rate. So this is the updated phylogenetic tree of genomes that are available on GISAID as of today. And if we look at the A.2 cluster now, it now has 11 genomes, including a sublineage A.2.1, which comprises 5 genomes currently. 3 of them are from India, from the state of Kerala. 1 of them is from Florida, and 1 of them is from the United Kingdom. So in summary, our analysis showed that genomes from 3 different countries actually form a distinct lineage A.2 of the monkeypox virus. And the A.2 lineage is distinctly different from the B.1 lineage, which is actually linked to the European outbreak or the majority of the 2022 outbreak of monkeypox. The A.2 lineage genomes actually have 16 genetic variations, which are not observed in any other lineages. And the time to most recent common ancestor, or the date of the origin of this A.2 cluster, is suggestive of a cryptic and a long duration of transmission of the virus, such that it probably went undetected for a very long time, beginning 2021, until probably awareness about the disease came into being. And cases associated with the A.2 lineage were either linked to travel to Nigeria, which is in Africa, or the Middle East, that is UAE. So now we know that computational methods for surveillance of different pathogens are actually very useful when it comes to understanding the evolution of the virus or the transmission dynamics of the disease. And this epidemiology of pathogens is actually heavily dependent on the downstream bioinformatics tools. We know that computational methods have helped us in early identification of positive agents of disease outbreaks such as COVID-19. They have helped us to estimate the origin of pandemics and develop public health measures. They are now helping us to trace the spread of disease outbreaks, monitor evolution of the pathogen, and track their prevalence. And we now want to move on to use all of this genomic data that is present in public domain to analyze all these big genomic data sets, unify the data, and come to some useful conclusions about how the disease is progressing throughout the world.
Bani,
However,
just
there are
to let
certain
you know that we're at time, if you could just wrap up quickly.
Sure, just last few slides. So there are a few challenges when it comes to informatics for monkeypox virus surveillance, such that computational expertise may be scarce in some regions and tools may not be easy to use for most researchers that are handling genome sequencing. And to remove these roadblocks, in the case of the SARS-CoV-2 pandemic, we had actually developed a tool known as the Genetic Epidemiology in a Box, or GISAID box, which is a tool that automates analysis and assembly of SARS-CoV-2 genomes and perform analysis related to epidemiology, such as automatically assigning lineages, creating next strain builds or phylogenetic builds, and submitting the data to GISAID. So we now just want to leverage whatever we have learned from the past from the ongoing COVID-19 pandemic, including all computational methodologies, surveillance tools, and so on, so that we are better prepared for other infectious diseases that may emerge or are emerging currently. So with that, I'll stop and I'll thank firstly my supervisor, which is Dr. Vinod Karia, and all the investigators from across the world that have deposited genomic data that have made all these analyses possible, including CDC USA and the National Institute of Virology in India. And also, of course, the funding agencies, which is CSIR India, which has made all these analyses.